This is a special recording to talk about substance P specifically and its role in the processing of pain. What is substance P? It's a neuropeptide. P in substance P stands for preparation or powder. Substance P is a peptide that's made of 11 amino acids. Okay, its concentration is dense, greater in the dorsal horn of spinal cord, substantia nigra, and amygdala. It works closely with many neurotransmitters, norepinephrine, serotonin, glutamate are a few mentioned here. And it is released from sensory nerve terminals in the skin, muscle, joints, then rapidly inactivated when it's not needed, and is important in inflammation. It promotes vasodilation with the help of nitric oxide, promotes cell growth, cell division, and it's linked to nausea and vomiting. The center for nausea and vomiting is in the medulla oblongata, and substance P seems to have a role in it. In addition to this, substance P is also important in the bronchoconstriction. Okay, now substance P binds to a receptor, okay, called neurokinin 1, NK1, which is expressed in on the cell surface. You know why? Cell surface, because substance P cannot make its way into the cell. A peptide being bigger is restricted from entering the cell membrane because of its large size and requires a receptor on the cell membrane facing the outside, and that's where you would find NK1. Okay, there are subtypes of NK1, but we don't have to know all that. G protein coupled receptor mechanism, GPCR, is G protein coupled receptor mechanism, is what is turned on when substance P binds to NK1. Okay, once G protein is activated, there are many mechanisms that can be turned on in the target cell, but here specifically phospholipase C pathway is turned on. And now what it does is the phosphorylation of subunits of the NMDA receptor. Okay, NMDA receptor, when it's phosphorylated, then it becomes an active receptor. Okay, this active receptor can then bind to glutamate and then bring about the effects of glutamate in its target cell. So what we should know here is that substance P does not really have a direct effect, but it simply enhances or facilitates the effect of glutamate. Okay, so that's how you have to understand the role of substance P. Okay, substance P would put the NMDA receptor from an inactive state to an active state. Okay, then glutamate binds to NMDA receptor, brings about its effect. Good. So substance P, when it's not needed, is degraded by its internalization by NK1 receptors and their target cells. NK1 is present in many places: spinal cord, hypothalamus, amygdala, and periaqueductal gray is where you see its distribution in the CNS. NK1 is present on many cells. That means substance P can bind to these cells and bring about its effect. Okay, neuron, neuroglia, endothelial cells. Vasodilation is where it has to bind to endothelial cells, for example. Fibroblasts, lymphocytes, stem cells. Lymphocytes is where substance P can bind to and trigger the release of specific chemicals called cytokines. Okay, now stem cells and white blood cells. And now cytokines can induce NK1 independent of cells can induce NK1 independent of substance P. That means cytokines directly can activate NK1 receptor and bring about the effect almost like substance P. And now I want you to see there, substance P can induce the release of cytokines or activate cytokines, which can then induce NK1. So basically, it's the paracrine effect that you see. I activate you, you activate me, and we both activate the target cell kind of approach is what is used here. Okay, good. Now, substance P and its role specifically in the processing of pain. Okay, so substance P is the first responder to pain stimulus, coexists with the glutamate. We'll look at some slides that would show that. Then it reduces pain threshold so that the second order neuron can now carry the pain signal forward. Okay. Now, two approaches we have looked at so far. One is substance P activating an NMDA receptor so that glutamate can bring about its effect. Secondly, substance P can lower the threshold of the second order neuron so that the action potential can be generated faster. Okay, amplifies excitatory responses to glutamate in the dorsal horn. Now, substance P can diffuse farther from the site of release to reach receptors at remote targets and regulate synaptic transmission at many different neurons. All right, so now let's talk about the details in the dorsal horn. What you're looking at here is the first order neuron carrying pain signal. And now what you see is the release of substance P. And you have NK1 receptors here, NK1 receptors in lamina 5 and other places as well. So that's a really neat demonstration of how there is this release of substance P in the dorsal horn by the pain stimulus. Okay. Good. With that, let's move on to talk about another illustration here. You're talking about first order neuron or primary efferent neuron carrying the pain signal. Now, I do not want you to worry about all these abbreviations, okay? Focus on glutamate, focus on substance P. Okay, so both are made by the same neuron, okay, in response to pain stimulus. And then glutamate, what it does is released. It binds to its NMDA receptor and AMPA too, AMPA receptor. Now, substance P is released as well. It binds to its receptor NK1. Okay, so both glutamate and substance P are sensitizing the postsynaptic cell, which happens to be our second order neuron, okay, projection neuron, right? So now what you're looking at here is the presence of nitric oxide, and these are inhibitory interneurons. You see what I mean? So here, one inhibitory neurotransmitter is here, and then nitric oxide released, and it binds to nitric oxide sensitive guanylin cyclase unit. Okay, it's a subunit. And then that would lead to the release of cyclic GMP, and that causes sensitization. Okay, so now you're talking about loss of inhibition. So you're talking about nitric oxide. Nitric oxide is another inhibitory interneuron. Okay, so this is where we are. This is a good understanding of how complex the mechanism is for substance P, glutamate, and first order neuron, second order neuron, inhibitor neurons, nitric oxide, just a complex arrangement, okay? So this is another nice illustration. I want you to pay attention to the response here. You're talking about glutamate in substance P. Okay, glutamate is here. Okay, and it has a certain response. Okay, take a look. Substance P increases excitability of glutamate. So this is just glutamate. When you add substance P, okay, you're infusing substance P, the response is increased. Got it? Glutamate by itself can work, but it is enhanced by the presence of substance P. That should be a good way to basically end this. Okay, good. Thank you.